So going to the next part, that is the renal tubule in details, we can say that it is consisting of three parts: PCT, loop of Henle, DCT, like I have already discussed. So the PCT is the proximal convoluted tubule. Because of its tortuous path, it is called like this, convoluted tubule. Now it is comprising of simple cuboidal cells, and it has prominent microvilli on the surface. Like you can see, this is the inside of the PCT. So there are microvilli, finger-like projections. Because of this, the surface area has increased and it is allowing for maximum absorption. So it is also secreting certain solutes such as sodium, chloride, glucose, etc. So the most important function of this is the transport of these ions across the membrane, and they possess a high concentration of mitochondria because they actively transport ions. Active transport means that's involving ATP, so that it can produce sufficient amount of ATP. So they have a high number of mitochondria in this part of the renal tubule. Now the PCT maintains the electrolyte and acid-base balance of the body fluids. Next is the loop of Henle. As you can see that it is a U-shaped tube and it consists of a descending limb and an ascending limb here. So it has a thick descending limb first, short, and it is going and it is forming a thin descending limb, then it is going up forming thin ascending limb, then it is again going up forming thick ascending limb. So of course it has a descending limb, ascending limb. Now it transfers the fluid from PCT to DCT, and they run parallel to each other. The two limbs of the loop of Henle, they run parallel to each other. They make a hairpin turn at the deepest point in the renal medulla, and then again this like I already said, there is an initial short thick portion, then a long thin portion, then again the ascending has a short thin portion and followed by a long thick portion. So the descending limb is highly permeable to water, but it is completely impermeable to ions. So therefore a large amount of water is reabsorbed over here, and therefore the fluid osmolarity is increasing. Okay, now in contrast, the ascending limb is impermeable to water, and therefore it is highly permeable to ions, and therefore it causes a large drop in the osmolarity of the fluid, because it allows that the water is reabsorbed inside the body from this urine in the descending limb, and then again in the ascending limb it's highly permeable to ions, therefore there is a large drop in the osmolarity, the ions also go out. So the DCT, the distal convoluted tubule, like the PCT, is also very much convoluted and is formed by simple cuboidal epithelium only, but like we can see that there are fewer microvilli on the surface, and they also pump ions against the concentration gradient. Therefore there is active transport of ions, so there is a large number of mitochondria here, but also the number of mitochondria is lesser than in the PCT. And it is shorter in length than the PCT, and it also secretes ions such as H+, K+, ammonia into the filtrate. So there is some secretion of ions and solutes in the membrane, in the DCT, and the filtrate, it also reabsorbs HCO3- from the filtrate. Now there is conditional reabsorption of sodium ions and water that takes place in the DCT, therefore it maintains the pH and also the sodium potassium level in the blood cells. Now the collecting ducts. The collecting ducts, as I have already mentioned, it's technically not a part of the nephron. It's continuous with the nephron, where all the DCT of the nephrons, surrounding nephrons, they come and they merge together in the collecting duct. So they descend deeper in the medulla. They form about 30 terminal ducts. Now the collecting ducts, they also go and form the merge in with the terminal ducts. They empty at a papilla. So they are lined with simple squamous epithelium like this. They have receptors for ADH, the antidiuretic hormone, like I have already told you about. And when stimulated by the ADH, these cells, they insert aquaporin channel proteins into their membranes. These are nothing but water channel proteins. They allow water to pass through the duct, and therefore from the duct it moves into the interstitial spaces in the renal medulla, and from there it goes into the vasa recta. Vasa recta is nothing but the network of capillaries surrounding the renal tubule, the peritubular capillaries. So Mostly the cells contain aquaporin molecules. They are so small that only water can pass through them, and like the name suggests, they are known as water channels as well. So positive charges, so this is the structure of an aquaporin. There are positive charges inside the channel. These charges prevent the leakage of electrolytes across the cell membrane, while allowing the water only to move through via osmosis. So the collecting duct, like I've already said, it's a long straight tube where hydrogen and potassium ions are secreted to maintain the electrolyte balance of the blood. This is also the region where maximum reabsorption of water takes place to produce a concentrated urine. Okay, so the next and most important thing is the mechanism of urine formation. So there are three main stages that are involved in the process of urine formation. They are glomerular filtration or ultrafiltration, selective reabsorption, and tubular secretion. So glomerular filtration is nothing but that filtration of the blood which enters via the afferent arteriole into the glomerulus and then goes out via the efferent arteriole, and in between the process that takes place inside the Bowman's capsule by filtering the blood and taking out the waste substances, harmful substances, etc. from the blood, and it goes inside the Bowman's capsule. This mechanism is known as filtration, glomerular filtration. So it takes place through the semi-permeable walls of the glomerular capillaries and Bowman's capsule. Now the afferent arterioles is supplying blood to the glomerular capsule. They contain some useful substances as well as certain harmful substances. So the useful substances are glucose, amino acids, vitamins, hormones, electrolytes, ions, etc. And the harmful substances are metabolic waste such as urea, uric acid, creatinine, etc. So they definitely need to be excreted by the urine. So what is the main procedure, like what is the schematic? There is a primary urine, and then the filtrate, that filtrate passes through the PCT, which reabsorb useful substance from the primary urine. So whichever substances are useful to the body, the PCT will start reabsorbing from that urine. Now partial reabsorption of water and sodium ions that takes place from Henle's loop. Sodium and water is reabsorbed under the influence of hormone, that is ADH, in DCT. Then again in the DCT, the urea, uric acid, creatinine is secreted by the tubule, and then the final urine is formed. So if we can see here, this is A is the glomerular filtration, and water and solutes that are smaller than proteins are forced through the capillary walls and pores of the glomerular capsule into the renal tubule. Next is the tubular reabsorption, B, where you can see that from the PCT, the capillaries, it is entering into the capillaries. What is entering? Water, glucose, amino acids, and the needed ions. Whatever useful that is there in the filtrate over here from the PCT goes back to the body again via the capillaries. Now then again there is tubular secretion. Some substances that are harmful but are not removed, they are getting secreted inside the PCT or the tubule, renal tubule, from the peritubular capillaries, from the blood of the peritubular capillaries. So these three are the major events taking place in the formation of urine. So what are the factors affecting the ultrafiltration? So first of all, what is ultrafiltration? Ultrafiltration is basically what is happening in the Bowman's capsule. Like there is a dense capillary network called glomerulus, where blood is flowing through afferent arteriole, carrying over through efferent arteriole, and there is a blood pressure difference between these two arterioles. Why? Because the renal artery, it contains blood at a very high pressure. So this is the short afferent arteriole, and the efferent arteriole has a smaller diameter, like you can see, that is narrow. So therefore there is high resistance, and this short wide afferent arteriole has low resistance pathway. So of course the blood will gush into the glomerulus and it will come out very slowly because of the narrow efferent arteriole. So blood will stay here longer, and therefore a high pressure will be created inside the glomerulus. So this high pressure forces small molecules such as water, glucose, amino acids, sodium chloride, urea, they go from the blood to the glomerular capsule or the Bowman's capsule, the empty space over there, the Bowman's space. This type of high pressure filtration is known as ultrafiltration. So the fluid formed here is called the glomerular filtrate. And the plasma proteins and the blood cells, they do not go inside because they are too big to be filtered. So in the glomerulus, now 3 main pressures are acting upon this filtrate. One is the glomerular blood hydrostatic pressure that promotes filtration, that is favoring the filtration. It pushes water and solutes in blood plasma through the glomerulus. So this is the blood pressure in the glomerular capillaries. It is around 55 mmHg. Next is the capsular hydrostatic pressure. There is a back pressure. It is opposing the filtration. So as the filtrate is forced into the capsular space, it meets 2 resistance. One is the wall of the capsule, that will obviously give a resistance, and the other, that is the fluid that is already present in the renal tubule, that will also create a resistance, like it will not allow any further liquid to come away so easily. So as a result, there will be a pushback. There will be a pressure which will be pushing back the filtrate from the glomerulus. So that is called the capsular hydrostatic pressure. Next is the blood colloid osmotic pressure. That is the second force that is opposing filtration. It is mainly due to the proteins present in the blood like albumin, globulin, etc. So the greater the concentration of solutes in a solution, of course, the greater is the osmotic pressure. So because blood contains a much higher concentration of solutes, its osmotic pressure is also high than the glomerular filtrate. So some water will move from the filtrate back into the glomerular capillaries, because water will always move from a region of hyperosmotic condition to a region of hypoosmotic condition. So from region of hypoosmotic condition to a region of hyperosmotic condition. So some water will move from glomerular filtrate towards the blood. So this is on an average, this pressure is around 30 mmHg. Now what is the net filtration pressure? So the net filtration pressure will be whatever there is the glomerular blood hydrostatic pressure and minus whatever capsular hydrostatic pressure is there, whatever the pressure that is exerted from the walls of the capsule and the filtrate already present in the capsule, and the third, that is the blood colloid osmotic pressure, which is the pressure of the osmotic concentration of the blood that is causing certain water to flow from the filtrate to the blood. So that pressure will also be subtracted from this glomerular blood hydrostatic pressure. So NFP will be equal to GBHP minus CHP plus BCOP. That is coming around 10 mmHg given the average values of pressure, whatever is given here, that is coming around this. So this means that only a pressure of 10 mmHg can cause a normal amount of plasma to filter from the glomerulus into the capsular space. Now GFR, that is the glomerular filtration rate, is the amount of filtrate formed in all the renal corpuscles of both the kidneys per minute. So that is the per minute, whatever the amount of filtrate, volume of filtrate is found, that is the glomerular filtration rate or GFR. In normal adult, it is about 125 ml per minute, which amounts to about 18 liters per day. Now GFR will obviously be affected by whatever the pressures of NFP or whatever the factors that affect NFP. Now this NFP is any factor that alters NFP will affect GFR, that is severe blood loss reduces systemic blood pressure, which will also decrease this glomerular blood hydrostatic pressure, and if this drops down to 45 mmHg, then the filtration will be stopped, because 45 minus 45 will become 0. So there will be 0 pressure and therefore there will be no filtration. There will be no net filtration pressure. So no filtration will happen, and therefore in this condition the urine output will be very less, like less than 50 ml per day, and that condition is known as anuria. So this homeostasis of the body fluids is maintained by the kidneys because of this GFR, because of this net filtration pressure. If the GFR is too high, like all the useful substances from the blood is also getting carried away by urine, then we have to lower it down. On the other hand, if it is too low, then what will happen? All the filtrate may be reabsorbed and certain waste products will also stay in the body only and they will not be properly excreted. Then that will also be detrimental to the body. So going into the next part of the urine formation, that is the selective reabsorption. So what is selective reabsorption? As the filtrate passes to the renal tubules, some useful substances like water, electrolytes, nutrients such as glucose, amino acids, vitamins, hormones, they are again reabsorbed from the filtrate back into the blood in the PCT, either via active transport or via passive transport. So either utilizing ATP or without utilizing ATP also. So a major portion of water is reabsorbed by osmosis. So only 60 to 70% of the filtrate, it reaches the Henle's loop. From there also it is reabsorbed and only 15 to 20% reaches the DCT. So as the filtrate flows through the descending loop of Henle, only water is reabsorbed and sodium and other solutes are not absorbed here. Further, when it goes into the ascending loop of Henle, the reabsorption of ions takes place because the loop of Henle, ascending limb, is impermeable to water. So no water is reabsorbed here. So the filtrate then flows through the DCT where active reabsorption of sodium takes place. So sodium is uptaken from here. Now the DCT is also under the, when it comes under the influence of aldosterone, then sodium is uptaken over here. Water is reabsorbed under the influence of ADH. Okay. Now aldosterone and ADH will act upon here and sodium and water will be reabsorbed from the DCT. Now the main function of the collecting ducts is to reabsorb as much water as possible because body needs water. So you cannot let go of the water in the form of urine. So you have to reabsorb the water and take it back into the body again. Now glucose, amino acids, vitamins and positively charged ions are reabsorbed by active transport while negatively charged ions are reabsorbed mainly by passive transport. Small proteins are absorbed by pinocytosis. So this is selective reabsorption, and just the reverse is tubular secretion. So some substances also needs to be secreted into the tubule, inside the tubule, so that it goes and excreted by the urine. Tubular secretion takes place from the blood in the peritubular capillaries to the filtrate in the renal tubules, which ensures that certain wastes which are present in the body such as creatinine, excess H+ or K+ ions, they need to be excreted. So they are tubular secretion makes sure that they are excreted from the body with the help of urine. So removal of H+ ions is essential for maintaining the pH of the blood. Also removal of excess K+ ion is essential because otherwise it will lead to a condition called hyperkalemia. So in exchange of sodium ion, excess K+ ion is secreted in the tubules. K+ is given out, sodium is reabsorbed. So certain substances such as drugs like penicillin, aspirin, they may not be filtered entirely out of the blood because of the short time given in the glomerulus. So what happens is that they are secreted into the tubular fluid, tubular filtrate, and by the capillaries, and therefore the filtrate carries it away from the urine with the help of urine. Secretion is the final step in the formation of urine. Certain substances such as creatinine, hippuric acid, drugs, etc. are actively secreted into the filtrate in the PCT. Urea also enters the filtrate by diffusion in the thin segment of the ascending loop of Henle, and then H+, K+ ions are also secreted into the filtrate in the DCT and like here, and therefore this final tubular filtrate is known as urine. This urine is obviously usually hypertonic because most of the water have already been reabsorbed by the body and taken back. Now there is a very important mechanism called the counter current mechanism. So what is the counter current mechanism? It is basically the process of creating an osmotic gradient, and it helps to reabsorb water from the tubular fluid and to produce a very much concentrated urine. So what happens is that in the loop of Henle, like I've already said, the descending limb is permeable to water. but not solutes, and the opposite is happening for ascending limb. It is permeable to ions but not permeable to water. It is impermeable to water. So also this loop of Henle goes inside the renal medulla. So renal medulla is very much highly concentrated in sodium chloride concentration, in lot of salt concentration. So it is hyperosmotic. So therefore this hyperosmotic environment will obviously take up water from whatever hypoosmotic solution is there. So this happens when this filtrate is passing through the loop of Henle. Because the two limbs of loop of Henle perform two different functions, it acts as a counter current multiplier. And the vasa recta, that is the tubular capillary surrounding the loop of Henle, it acts as a counter current exchanger. So water flows from the filtrate to the interstitial fluid, and therefore the osmolality is decreasing inside the descending limb. It increases because first, suppose it is 300 milliosmol, then it is going down to 600 because the water is getting out from here. Then it is getting concentrated, it is becoming 900. More water leaves the filtrate, it is becoming 1200. So it is highly concentrated at the bottom. This is highest concentration of urine is found here at the bottom point of the loop of Henle. Now what happens? Water is impermeable in the ascending limb of loop of Henle, but solutes, the solutes are permeable. So it is permeable to the solutes. Now what happens will the sodium and chloride ions will exit through the channels, like here. Sodium, chloride, potassium, that is exiting. Now blood is again, so the filtrate is again coming towards 900, 600 and 100. It is becoming dilute constantly. So it is active, so the ions are actively transported out of the filtrate, and therefore by the time the filtrate reaches the DCT, most of the urine and solutes have been reabsorbed. If the body requires additional water, all of it can be reabsorbed at this point. So the filtrate that has entered has becoming progressively more concentrated as it loses water, and then blood in the vasa recta removes the water from the loop of Henle, because this water is coming out in the interstitial fluid and the vasa recta is taking up this water from the interstitial fluid only. So here the vasa rectas, here it will become dilute by taking up water and going up. And when it is coming down, it will be becoming concentrated because it is getting ions from this side of the loop of Henle. So the ascending limb is pumping out sodium, potassium, chloride and filtrate becomes hypoosmotic. It is becoming dilute. So this happens because there is differential permeability of the two limbs of the loop of Henle. Now this counter current mechanism, there are three segments in the loop of Henle, they have different characteristics. The thin descending limb is passively permeable to both water and small solutes, like I have already said, and therefore the tubular fluid becomes steadily more concentrated or hyperosmotic as compared to the blood as it travels down the thin limb of the descending loop of Henle. Now the thin ascending limb is passively permeable to small solutes, but it is completely impermeable to water. Since it is impermeable to water, therefore there is passive movement of sodium chloride ions from here, and then as it going up in the thick ascending limb, there is actively reabsorption of the sodium chloride ions. And here the urine is again becoming dilute. So this is a diluting segment. Okay, so one is the filtrate entering, it is isoosmotic to both plasma and interstitial fluid. Now water moves out and it becomes highly concentrated, hyperosmotic. Now again the filtrate reaches its highest concentration at the bend of the loop. Then what happens? The sodium and chloride are pumped out, it is becoming hypoosmotic again, and then the filtrate is at its most dilute as it leaves the nephron loop, and at 100 milliosmol it is hypoosmotic to the interstitial fluid. So this is the counter current mechanism. First it is getting highly concentrated, then it is again getting diluted. Now from here the water can again be reabsorbed by the body if the body is dehydrated. So coming to the composition of urine. So urine is basically a liquid or a semi-solid kind of solution, because it's comprising of the waste coming from the metabolism activities of the body, and it also contains certain harmful compounds that needs to be excreted from the body. So urine contains both organic urea, creatinine, uric acid substances and inorganic substances like sodium, potassium, chloride, ions. Now the presence of proteins in urine gives it an acidic pH of around 6. So in urine mostly 95% is water and the rest are ammonia, phosphate, magnesium, potassium, creatinine, urea, uric acid, sodium, calcium, chloride, sulfate. So urobilin, this is basically the pigment which is present in the urine. It is a byproduct of the breakdown of heme from hemoglobin during the apoptosis of blood cells. So the process of time to time collection and removal of urine from the urinary bladder is known as micturition. So the release of urine from the urinary bladder and time to time urination is known as micturition. So the collection of more than 300 ml of urine in urinary bladder, it creates a pressure on the walls of the bladder, and therefore this pressure stimulates or causes the urge for urination. So how this micturition reflex works, it actually this is the stretch receptor in the urinary bladder. So this stretch receptor goes and sends a signal via the afferent neuron to the parasympathetic or to this pelvic nerve, and it goes through the spinal cord, and this interneuron relays the sensation to the thalamus, and here the projection fibers from thalamus deliver sensation to cerebral cortex, and therefore this interneuron again comes back with an efferent message, and here it is message is transferred to the bladder, like the stimulus the contraction of the muscle. So when it triggers the contraction of the muscle of the urinary bladder, then the urine is released and by urination. So after micturition occurs after voluntary relaxation of the external urethral sphincter, then only in urination can occur. So this is the basics of the excretion chapter, and we have completed the unit 5 over here. Thank you.